Okay, folks, we have the luxury of actually running a few minutes ahead of schedule, but I think we're going to get started with this final session of the meeting. I see many of you have found the box lunches out in the hallway, so if you haven't, please feel free to pick one up. So again, I'm Rich Schilsky from ASCO, and this last session of the meeting really is intended to focus on a number of somewhat diverse regulatory issues that many of us doing clinical trials have dealt with over the years, some of which impact on the efficiency of completing clinical trials, and we thought we would highlight a number of these areas this morning and then have some discussion about how to achieve the regulatory goals and doing so in a maximally efficient way. So actually, I'm going to start off by talking briefly about a project that Jeff Abrams and I and a few other people were involved in a few years ago now regarding optimizing the collection of safety data in supplemental clinical trial applications, so applications seeking supplemental NDAs or BLAs. This is an area that has caused a lot of consternation over the years, in large part because oftentimes sites are buried in a blizzard of safety reports, many of which turn out to be largely irrelevant to evaluating the actual drug product or patient population. So what are we talking about here? We're really talking about establishing data collection standards that meet clinical needs, regulatory standards, as well as the goals of cooperative groups and industry. We're talking about trying to define a minimum data set that's necessary to support a claim of safety in a supplemental application. And again, I want to emphasize that throughout this presentation, I'm talking about supplemental applications. So the implication of that, of course, is that there already is an approved marketing application for a drug. That is, the drug is already on the market for some indication. And now we're talking about the collection of safety data for a new indication. So we're talking about specifying the data required to permit appropriate labeling and to inform clinical use in this new indication, clarifying the nature, extent, frequency, and format of clinical data reporting, and determining the percent of the study population in a clinical trial that is actually necessary for informative data capture, and whether or not one could actually limit the amount of safety data collection to a subset of a population in a large clinical trial. Why is this important? Well, it's important to ensure adequate data collection to inform regulatory and clinical decisions. It's important to protect patient safety by improving the overall quality of data that's submitted in supplemental drug applications. I think many of us who either have been involved in coordinating clinical trials or on the front lines of performing clinical trials in our institutions or practices realize that there's so much information that is often required by a study sponsor to be submitted that we have a forest and trees approach. And sometimes the important safety signals can occasionally be obscured by all of the additional information that is being required that oftentimes is not terribly informative. So we want to also, in assessing this, try to increase the efficiency of clinical trials by eventually having the resources to develop more drugs with the limited resources that are available. We want to reduce the data collection burden on the clinical trial system, aligning the resources to focus on the key data elements that are actually necessary to achieve regulatory and safety goals. In doing so, we hope to be able to actually enhance physician participation in clinical trials. And one of the big obstacles we hear from doctors all the time is that there's so much paperwork involved in doing clinical trials that it's just not worth the trouble. And if we can reduce the paperwork, if you will, to the minimum necessary to achieve the scientific and regulatory and clinical goals, maybe we'll get more doctors involved. And maybe we'll reverse the trend that's been prevalent for many years now of new agents being studied outside the United States because of concerns about just the amount of effort and expense required to meet the requirements within the U.S. So the general principles that we've been considering in this whole project, which is now completed and published, so I'm not going to go through it in great detail because you can look up the JCO publication of a couple of years ago, and we can certainly provide that. But the guiding principles really are to collect the necessary data to inform regulatory review, labeling, and clinical use. That the data that is collected in clinical trials should actually be used for some purpose, and that we shouldn't be collecting data that is not used or not useful. That data collection for new drug applications should remain comprehensive. That's new drug applications. But the data collection requirements for supplemental applications could vary based on the safety database and known pharmacology and drug interactions for the agent under consideration, the similarity of the study population in the supplemental application to the original study population when the drug was first approved, the similarity of the regimen being studied in the supplemental indication to that already approved, and whether or not the supplemental application follows an initial full approval or accelerated approval, essentially relating to the size of the existing safety database about the agent under consideration. The feeling was that reduced data collection would apply only for agents with a well-defined safety profile that had received regulatory full approval. So the implication again being that there was already a substantial safety database available about the drug, and that for supplemental applications that we would collect the necessary safety data to inform regulatory review, labeling, and clinical decisions. And that would include symmetric data collection across all the arms of the study, collecting detailed information on study deaths and serious adverse events as traditionally defined, collecting data on adverse events that are clinically meaningful, that is, those adverse events that actually lead to discontinuation of a drug or dose modification of a drug because something bad has happened to the patient as a result of taking the drug. And then we would collect targeted adverse events and information on concomitant medications as needed based on the drug's known safety profile or pharmacological class. So, for an anti-angiogenic, you might want to be sure to collect data on hypertension or cardiovascular events, but you wouldn't necessarily do that routinely for every drug, every drug class that perhaps is not known to have any particular effects on the cardiovascular system. So that's all good and well. The question then became, well, can we come up with any data to sort of back up these general principles and demonstrate that, in fact, we would not be missing important safety signals if in some way we reduce the amount of data safety collection in supplemental applications. So under the auspices of ASCO back in 2008 now, a group known as the Data Optimization Working Group was formed. And its charge really was to reanalyze multiple clinical trial toxicity databases and examine various sampling methods to determine if optimized data collection would provide sufficient safety data to support supplemental applications. The objectives of doing this were to determine what adverse events might be missed through sampling a subset of trial participants, determine if there was a target subset size that minimizes the chance of missing clinically important adverse events, determine a subset size in which noise events are acceptably low, determine if there's a preferred method for subsampling patients, assess the extent of data collection and cleaning effort that would be saved if we could collect less safety data, and evaluate what concomitant medication data is collected and what is actually used in regulatory and clinical decisions. So to do this, we actually were able to enlist one cooperative group, the CALGB, and 4 companies, GSK, Eli Lilly, Novartis, and Genentech, in a project. And each of the sponsors there contributed data from one or more large prospective randomized clinical trials that had been used for regulatory submission. In advance of beginning the project, all of the participants agreed to and wrote a statistical analysis plan, so we could sort of prospectively specify the plan for undertaking this project. We submitted it to the FDA, not for a formal evaluation, but to get any comments that they might have informally. And then this statistical analysis plan was then the guidelines for doing the simulations that were done on the clinical trial data sets that were contributed by these companies. So each candidate clinical trial, we basically did a whole bunch of simulations looking at collecting various types of safety data in various segments of the study population. So groups of 200, 300, 400, 500, or 600 patients, usually in trials that enrolled 1,000 or more patients. So there were 8 studies involved in both the metastatic and adjuvant disease setting. And we were interested in learning what we would learn regarding grade 3 and 4 adverse events, as well as about grade 1 and 2 adverse events relating to what was previously known from the safety database already available for the study, what we learned from analyzing the simulations in the study, and then what was already available in the medical or clinical literature regarding the particular drug. So we collected detailed information on all study deaths and SAEs. We collected information on all adverse events leading to drug discontinuation or dose modification. That was part of our guiding principles. We focused on subsampling grade 3 and higher AEs that had at least a 2% difference in frequency between the treatment and control arms in the study. And for grade 1 and 2 AEs, those that had at least a 5% difference in frequency between the treatment and control arms of the studies. And then we constructed, if you will, or the context was the known safety profile of the particular drug that was determined from the original NDA filing, the drug label, the safety database available for the study, and the medical literature. I won't go through the details. These are some of the studies. You can see the contributing company, the study. These were studies in metastatic colorectal cancer, non-small cell lung cancer, and breast cancer. Several of the studies involved bevacizumab. One involved lapatinib, one involved pemetrexed. So it was a sort of a range of both biologics and cytotoxic drugs. You can see the sample sizes in these metastatic trials. The adjuvant trials, of course, were much larger and involved drugs like aromatase inhibitors and Herceptin, and you can see many thousands of patients enrolled in these studies. So what did we learn from this exercise? Well, we had toxicity records available for over 17,000 patients from these 8 trials. From the 17,000 patients, there were a total of 43 grade 3 or higher events that were observed in at least a 2% excess between the treatment and control arms. Although 34 of these events were already known safety signals for that particular drug. So there were a grand total of 9 events out of 17,000 patients that were observed in this process that might not have already been recognized. And for grade 1 and 2 events, the delta was 4. We observed 24 grade 1 and 2 events that were observed with at least a 5% greater frequency in the treatment compared to the control arm, although 20 of those 24 were already previously known adverse events. So a lot of safety data collected for a very little bit of incremental information. And this slide you can't read, but I'll summarize it here. So, as you might expect, as we looked at the size of the subset in the study, 200, 300, 400, up to 600 patient subsets, the rates of detection of grade 3 and higher AEs in excess range from about 60% to 100% in subsets of 200 to 600 patients. The chance of detecting an excess in the frequency of an adverse event increased with the rate of the adverse event excess. So, if it was an adverse event that occurred in 2% higher frequency in the treatment arm compared to the control arm versus 3% versus 4%, and so on. Obviously, the more frequent it was, the greater was your chance of detecting it, even in a smaller subset. For grade 3 plus AEs that occurred in at least 3% excess in the treatment arm, those were detected in 88% of all the subsets we looked at, regardless of size of the subset. And grade 3 plus AEs that occurred in at least 3% excess were detected in at least 75% of the simulations of a 400-patient subset. We're beginning to hone in on subsets of about 400 as probably an adequate sample size to detect most of the higher grade AEs that were likely to be clinically important. And in this slide, again, you can see just some examples, and I'll just highlight in red here. So on the left here, you have the subsets, 200 to 600 sample subsets, and across the top are various AEs. In the red box are the AEs that were occurring in these trials with 3% or higher frequency in the treatment arm compared to the control arm. And again, you can see then in the box, if you have a sample subset of at least 400 patients, at least 75% of the time, and in most cases, 80 or 90% of the time or more, you're going to be able to detect those AEs, even in a 400-patient sample subset. When we looked at some estimate of the effort that could be saved from reducing the amount of safety data collection. So again, it's a bit of a complicated slide, but you've got the studies, you've got the grade 1 and 2 events, the grade 3 and 4 events that were not considered to be events serious enough to require drug discontinuation or dose modification, and then the SAEs and the serious AEs, that is, in a sense, the ones that we defined as requiring drug discontinuation or dose modification. And just looking at the numbers, you can see that there are tens of thousands of events that are typically collected in these studies that essentially are non-informative and just create a lot of busy work. With respect to the CONMED data, and we didn't do a very formal analysis on this, but it was kind of interesting. And again, you can see that there are literally CONMED records that are collected on tens of thousands of patients in these studies. And CONMED data fields, meaning, what date was the drug started, what date was the drug stopped, and, what was the drug and what was the dose of the drug and so on that was administered. These are the concomitant medications that the patient received, not the study drug, just, anything else that they happen to be taking at the time. There are hundreds of thousands of data fields that our data managers are asked to complete in filling out these forms about concomitant medications. So in these particular trials that were included in this study, there were 136,000 CONMED records and many hundreds of thousands of more data fields comprising those 136,000 CONMED records that all had to be completed. It turns out that none of the CONMED data was incorporated into the labels for any of the drugs, when those drugs were approved. So essentially, if it wasn't incorporated in a label, it couldn't be clinically important or useful to physicians in guiding clinical management of patients. It was just completely unnecessary data collection. So our conclusions really turned out to be that reanalysis of data from these 8 clinical trials of varying type, duration, size, varying drug classes demonstrated that capturing excess grade 1 and 2 events did not appear to add to the known safety profile of the drug. that the probability of missing a previously unrecognized and clinically significant grade 3 or 4 adverse event was very low. The probability of adding a noise event was also low, and that similar conclusions regarding the safety profile of the drug would have been reached from the subset analysis as would have been obtained from a full data collection in these particular studies. Review of the CONMED databases from 6 of the trials demonstrated that no new information was learned from the summary tabulations that were listed in the SNDAs or SBLAs. And we believe that with regard to CONMEDs, that useful information is typically learned either from the initial clinical trials, SAE narratives, where a physician might actually clinically implicate a concomitant medication, potentially targeted CONMED data where you have a hypothesis that there could be a pharmacological interaction between a study drug and another pharmacological class of agents that patients might be receiving. So with regard to CONMEDs, our conclusion was that information on CONMEDs should not be collected in supplemental applications, except in certain circumstances. That could be continued to be reported in the narrative section of SAE forms. They could be collected in a targeted fashion based on a hypothesized known drug interaction with another class of drugs. They could be collected if they represent a class of drug that may itself have an anti-tumor effect that could confound interpretation of the primary endpoint of the trial. So, for example, maybe collecting data on bisphosphonates in an adjuvant breast cancer trial, or they could be collected if collecting them addresses a specific study endpoint. Other than that, there doesn't seem to be any good reason to collect them in the context of supplemental drug applications. So the recommendations from this study were that for future supplemental trials that fit the general qualifications that we put forth in this study, that it was not necessary to collect grade 1 and 2 adverse events, that it was not necessary to collect grade 3 and 4 events in all patients, that a subsample of about 400 patients provides adequate probability of detecting events with at least a 3% excess in the treatment compared to the control arm, that it was unnecessary to collect start and stop dates for AEs, because again, generally information that was not used. It was unnecessary to collect data on all concomitant medications. And our hope in doing this project, which, as I said, was published a couple of years ago now, was that it would help inform the development of FDA guidance on this topic. Now, in fact, last year, the FDA did issue 2 guidance documents that are relevant to this topic. So the first came out last February, is entitled Determining the Extent of Safety Data Collection Needed in Late-Stage Pre-Market and Post-Approval Clinical Investigations. So that is directly relevant to the topic of this data analysis, although this guidance applies across all therapeutic areas. It was not specific for oncology drugs. And then just in December, FDA issued guidance on safety reporting requirements for INDs and bioavailability and bioequivalence studies, which is again an updated guidance across all therapeutic areas and focusing primarily on new drug applications rather than supplemental applications. The February guidance, which is the more relevant of the 2, acknowledges that in some settings it may not be necessary to collect certain types of safety data, recognizes that excess data collection may have negative consequences, proposes the collection of all SAEs and collection of AEs that lead to drug discontinuation or dose modification and targeted laboratory tests. So we're somewhat pleased that many of our observations seem to have been incorporated into this draft guidance document. It also discusses circumstances when comprehensive safety data collection is needed and the types of data that should always be collected. And it goes on to say that targeted safety data collection may be appropriate when certain circumstances are met, when the safety profile of the drug is already well characterized from prior studies, when the AE type and frequency is similar across multiple studies, when the expected adverse event rates in the study population are likely to be similar to those observed in previous studies, And it again states that targeted safety data collection may be appropriate for post-marketing studies for new indications, studies required to meet post-marketing requirements, and large outcome studies. So again, I think those of us involved in this project were quite gratified to see this guidance and felt that it was consistent with many of our observations from this study. The guidance also says that types of data appropriate for selective collection include non-serious AEs that don't require drug discontinuation, routine lab monitoring, con meds, history and physical exam. It mentions the collection of data from a sample of the study population and the possibility of decreased frequency of data collection. And of course, it advises that there should be a pre-specified plan for selective safety data collection that should be included in the protocol and should be discussed with the agency in advance of beginning the study. The only conundrum we faced in all this, just to wrap up now, the February guidance concludes also that in an oncology setting, it states that grade 3 and 4 AEs, as well as grade 2 events that affect vital organs, should always be collected. That seems to be a little bit counter to what the rest of the guidance says, although acknowledging that this guidance is not specific for oncology products. But it also seems to be somewhat at variance with a 2001 guidance from FDA that I reference in this slide that states that in supplemental efficacy applications that propose a new use for an already marketed drug in a similar population, that additional data on grade 1, 2 non-hematologic toxicity and grade 1 to 3 hematologic toxicity may not be important and may not need to be collected. Now, that is an oncology-specific guidance. So just to conclude, I think we're making great progress here. I think we have strong data that supports the notion that for supplemental applications that there can be substantial reduction in the amount of safety data collected without putting at risk missing any important safety signals. I think that's been acknowledged in recent FDA guidance or draft guidance on this topic. There seems to still be a little bit of work to be done to harmonize some of the guidance documents that are out there, and I know ASCO has requested some clarification from FDA, and we're waiting for a response. But by and large, I think we're moving in a good direction here by focusing on collecting the data that's clinically meaningful and important to make both the regulatory and clinical decisions. So I'll stop there, and maybe we can come back to some of these points in the discussion. So next up, I want to introduce Rachel Sherman from the FDA Office of Medical Policy, who's going to give us a little bit of a discussion of FDA's perspective on data quality issues. Rachel.